Thank you, Dibrugarh University Pharmaceutical Science Department, Orubda, and others for inviting to share our science and philosophy journey. And this is a holistic medicine seminar. I thought that's why I should talk about holistic science. And in a holistic science, we talk about philosophy and science intercourse, mainly. Today I will be talking about interface between philosophy and science and how we have a nearly 25 years of scientific discourse journey. I'll try to compress everything in about 30 minutes to 40 minutes. This is our KaviKrishna laboratory. KaviKrishna means vision to action. This is a Kamrup mantra. Is coming from Vedic one chant called KaviKrishna. So first I'll go mainly to this, our topic today. I will first going to go talk about cancer, its problem, its philosophical problem. Why cancer is still a killer disease. Then we'll talk about how our Vedic philosophy can contributing. And then we'll talk about how we have been able to assemble scientific experimental data, our journey, right from my clinics in Sualkuchi Hajo that I started in 1994, up to having a lab in IIT Guwahati and having a capacity building. We have now, few of my students who have completed their PhDs. They're here right now, so I'll talk about their journey as well. But this should be a scientific journey. And I think for pharmaceutical science, this is a good idea that even though you're always studying, memorizing, we should appreciate or maybe hear some story about how science developed, how science grew out of questions. So I don't have to talk about too much about cancer. Everybody, every family has a story about cancer. One of us, within 5 individuals, one of us are already getting cancer in our body. It's so big problem right now, especially in Assam. And this is my story. What has medical oncology practice taught me since 1994? I have about right now 500 cancer patients. We have a medical app called the Healer app. We monitor them through a telemedicine program. So what is teaching me is that something is wrong about how we are managing cancer. The mistake is here we have somehow. We're considering cancer from a Darwinian evolutionary view. It's clonal selection. So the mutation and selection. Our therapy is targeting this mutated clones. Our therapy is the chemotherapy to target and kill the cancer cells. So we're hitting the cancer radiation, chemotherapy, targeted therapy, because we believe that cancer is all about the competing clones. As you can see here, in this tumor, the clonal competition, which was introduced back in 1960 by novels, it is the paradigm for cancer biology. But there is something else we can talk about. That cancer could be a cooperative system. Today, after about half an hour and 40 minutes of our presentation, I would like to convince you to think that maybe we have mistaken that Vedic philosophy, Indian philosophy can bring some insight to what is cancer. So at the root of this, the dilemma that we have whether cancer is a competing clones or a cooperative system is Darwin's dilemma. Those who are not familiar with evolutionary biology, I think you all heard about Charles Darwin, his natural selection theory. But he had a real issue with honeybees. The bees and ants, they have a social life. They're altruistic. Many of the bees colony, they sacrifice their fitness for the enhancement of the group fitness. So Darwin then tried to reconcile his theory with his book, The Descent of Man. He said maybe selection may also happen in group, but he didn't have an idea. In 1964, Hamilton came forward. Hamilton proposed the kin selection theory. He developed a mathematical model. In this modeling, the altruism is all about the group fitness. Kin selection is RB. R is this the relatedness of the genetic relation, the genetically how much the recipient of the altruism, delivery of the altruism, they're related. Hambanda, kin selection. B is the benefit that the recipient is receiving. And the C is the cost of the altruistic. If the RB is the higher than the cost of the altruistic, then the gene could be selected through this group selection. However, it is very hard to prove it at the cellular level because of this issue. That we still don't know what could be the cellular and molecular mechanism at the cellular level to select those kind of signaling molecules down from the evolution of life. As you know, life began about 3.8 billion years ago as a unicellular life. Can you explain this altruism? Can you explain this origin of life with the selection of, through the kin selection and then the most difficult part is this. If there is a kin selection, if there's a group selection, that's an altruistic sacrifice, his fitness for a group, there have to be a group identity. How can you conceive an idea of a group identity amongst a clone of cells? Cells doesn't have consciousness. They are not human beings or others. So how can they sacrifice their fitness for a group? We need a group identity. Darwinians and other molecular biologists were always rejecting any possibility of a group identity amongst the multicellular life. So how is it possible for one of my cells in my body to sacrifice its fitness for a group? That has been another dilemma. The third one is this, that the cooperation itself is against this dogma of survival of the fittest, because those who are cooperating, they'll be always overwhelmed by the corrupt, those who are cheaters. Cheaters' genes always prevails in evolution. So then, that's why even though the bacterial altruism has been documented for last about 10 years, it has not been yet accepted because of those lack of molecular mechanism mainly. So I came with a very different approach. I'm introducing here an idea. I wrote a book about this Jiva Upakara Tantra back in 2018, but I was introduced to this idea by some philosopher, also some tantric healer, and I wrote a book about all my journey in Kamrup and Bhutan. It's a 500 pages book for about my search for this Tantra. So at the heart of this Tantra is the Pancha kosha system. Those who knows Upanishad, they know that Upanishad seers divided our body and mind as a 5 kosha or sheets. One is the monomaya kosha, mind, annamaya kosha, all this anna, made of food. Then the pranamaya kosha, made of air and metabolism. Vijnanamaya kosha, buddhi, intelligence. Then anandamaya kosha, our soul. Jiva Upakara Tantra introduced the 6th kosha called Upakara kosha. It's come from this Vaishnava Tantra called Pancha Tantra. It was practiced in Hajo Hayagriva temple long time ago. The innovation in this Upakara kosha is that during stress in our body and mind, a transient power, a Shakti arises. Everybody can feel that when you have stress, sometimes you get extra energy to work hard. That could be your Upakara kosha. Our tantric healer job was to activate those Upakara kosha by Tantra mantra. So I like the philosophy of this Tantra, the Upakara kosha. It sounds like altruism, as if Upakara kosha, even there is power, is trying to sacrifice its own fitness to protect the Pancha kosha system. So this is an AI model, of this. You can see avatar koshas like Narasimha. Narasimha avatar, transient, very powerful. So there, if we add this avatar kosha, we can resolve Hamilton's dilemma. In Hamilton, in the group fitness, this thing C, but we need a defending power. What is the use of sacrificing if you don't have power? That is my question. If the ant or the bees, when they're sacrificing their fitness, their reproductive fitness, but they're already weak, they're worker. In the Vedic altruism or Jiva Upakara Tantra, one who's sacrificing the avatar is already very powerful. They had a defending power. Despite having defending power, their power is sacrificing fitness, own fitness for the group fitness. As Krishna or Ram, this avatar, they had immense power, but they sacrificed their power for the benefit of the entire group. So these are the philosophers to introduce, and this is the book written by my father. He was also a philosopher of Jiva Upakara Tantra. In this book, published in 1991, he wrote a poem, and his main concept was that if we have cancer, if we have any disease in our body will have some avatar kosha, avatar cells that can defend our body. And my job in 1994 was to actually search for those avatar cells, or I call altruistic stem cells. So, one of the very interesting part of this Tantra is a Satavada Tarka, or the suppositional reasoning. As you know, India is famous for Nyaya Shastra, logic system. This Tantric, they used to have this Tarka system to debate with other Tantric, to also debate with ghost. Anybody who has seen a Tantric, sometimes they will have a dialogue with the ghost. So they will use this kind of Tarka. In Tarka, the initial thing is Ishcha Shakti. Then the protection, observation, Anuman, inference. Then Yukti or reasoning. Then Samasya, what is the problem? How to solve it? Then Dharana is an idea. And then it can lead to knowledge emergence. I learned this Tarka system during the Hambai Bab's Gita Shivir back in 1984. And also subsequently with Dal Krishna Bora, he was a Sangha Prasarak of Rashtriya Sangha Sangha. So it really helped me for scientific reasoning. So with that, this is actually symbol iconography in our Hatisatra and Sualkuchi, how this Satavada Tarka is a dialogue between Purusha and Prakriti, or Shiva and Narayan. Those are already iconographic, philosophical kind of achievement of our Kamrup culture or philosophy. I would not talk about that too much here. So there I depicted everything in a book I published in the year 2000, Science Behind Squalene. I'll talk about what is squalene later on. But you can see I mentioned this avatar kosha as one word because the editor of this book at the time was from Toronto. He did not allow me to introduce the Jiva Upakara Tantra. He only allowed me to use one word, avatar kosha. It was thought that, Bikul, why are you using a very religion for science? So that's why I had to omit entire paragraph. Only I kept avatar kosha. Suffice to say that this is the book where I first introduced that tantric idea of avatar kosha. So the squalene, as you know, all the pharmacological science students will know, this is a product of mevalonate metabolism, the cholesterol biosynthesis. Squalene has been in Earth since 2.5 billion years ago. We know isoprenoid fossil has been found. This is a cell membrane constituent. This lipid is the prime cell membrane constituent of archaeas, also cyanobacteria. This is also present in our skin. So what I proposed at the time in this book was that we had these ancestral stem cells during this Precambrian era, about 1.8 billion years ago, when unicellular life dominated this earth, those unicellular cells or ancestral stem cells, they had a defense mechanism, and probably they had an altruistic defense mechanism. And I propose that in our body, our hematopoietic stem cells, our mesenchymal stem cells, even embryonic stem cells, we can recapitulate, reactivate that altruistic defense mechanism. That we can demonstrate that altruism in vitro in a laboratory if we can smartly design experiment. For next about 10 minutes, I will talk about our experimental design, our challenges, and how we accomplished some success to demonstrate altruism by our stem cell and reactivating this ancestral stem cell defense as shown in this figure here. This is the ancestral stem cells model, and this is the stem cell defense. So this is my summary slide so far. Philosophy and my clinical experience. In my clinical experience, I always am surprised that even though we give chemotherapy to the cancer patients, very toxic chemotherapy, their stem cell recover, their blood counts goes up. After cisplatin chemotherapy for oral cancer patients, for example, the blood count goes down. The patient develop anemia. Why? Their bone marrow stem cells are targeted by our chemotherapy. Doctors sometimes stop the chemotherapy. Sometimes we have to give blood transfusion. It's because the chemotherapy drug that we are targeting cancer cells, they also target stem cells in our bone marrow. But eventually bone marrow stem cells recover. Many patients survive. Cancer disappeared. I always had the question, so that means stem cell has something. They have higher power than the cancer cell. So I gave a talk about this, my putative altruistic stem cell model, the University of Leiden in Netherlands. This is the picture. I was in the middle. I was very tiny at the time for the group. That was my first talk about altruistic stem cell as a proposition back in the year 2000. And then I also proposed in this book that if this is the case, if I design an experiment where we treat hematopoietic stem cells with cisplatin chemotherapy in the lab, that stem cell should exhibit altruism. This experiment we did, using this Satabra we designed the experiment. I have a student here, he's an oral talk to there tomorrow, Rupam Das. He's doing PhD in IIT Guwahati under me, and also Professor Uday Dixit. He's going to further explain what is Satabra Tarka, how we design experiment, scientific experiment using this Tarka. But using this Tarka, we designed an experiment, and this is the data we published in the year 2003. As you can see, after cisplatin treatment, this is the control of the hematopoietic stem cell colonies. This is after the cisplatin treatment here. And this is after we add some squalene in the culture media. The cells keep on growing, the stem cells recover. We can obtain some big cells, you can see these pink cells. We thought those are altruistic stem cells, but back in 2003, our technology was not well developed. We could not identify the stem cell marker. We could not further demonstrate what could be the molecular mechanism of those putative altruistic stem cells. That's an example. Sometimes you get some result, you know it may be, but you don't have the technology to prove it. It was back in 2003. And then we designed another experiment. This time we use hypoxia reoxygenation. Those who know about Earth, our primitive Earth was hypoxic. There was no oxygen 1.2 billion years ago. There was no oxygen. There's intense radiation only. In the toxic environment, Earth started developing oxygen because the bacteria was producing oxygen, bacteria and rocks. So we had a period of about 500 million years of hypoxia reoxygenation. Hypoxia reoxygenation. That generates reactive oxygen species. And I thought that our ancestral stem cell might have developed coping mechanism, altruistic defense against intense hypoxia reoxygenation. So let us simulate that microenvironment in the lab. So in this model, we expose stem cells, human embryonic stem cells, to this intense hypoxia for 24 hours. 0.1% oxygen only, there's almost no oxygen. After 24 hours, we then reopen this chamber, full oxygen. So with that kind of simulation, we wanted to find out if they have an activation of this ancestral stem cell defense mechanism, ancestral altruistic defense. And what we found, first using cancer cells as a model first, back in 2005, we discovered a molecular mechanism called VEGF receptor 1 MAP kinase pathway. We discovered an autocrine loop. In this loop, we have this HIF1, which is about 1 billion year olds. Phylogenetically, HIF genes, hypoxia-inducing factors, at least 1 billion year old. FLT1 or VEGF receptor 1 is equally very old. So we also MAP kinase ARC1 are very old system. Highly conserved molecular pathway for mitogenic signaling. So we discovered that pathway. It was a very one first step ahead that we may have landed in a molecular pathway for altruism. And then also what we found was that in 2008 we published in Oncogene with my Japanese collaborators that when you add cisplatin to cancer cells, this cancer cell activate this pathway, VEGF FLT1 MAP kinase pathway. They become kind of altruistic. That is the first hint back in 2008 that our chemotherapy drug may be actually helping certain cancer cells, cancer stem cells, to activate altruistic defense. That was the first hint. And then we have, with all those data from 2003 to 2008, we reorganized our experimental plan. We conceived that P53 MDMT could be a very important player here. Because VEGF, FLT1, HIF all regulate P53. I think everybody knows about P53. It's a guardian of genome, a transcription factor that activated to induce cell death or cell cycle arrest. If stem cells have any genomic damage, any DNA damage that activate P53 system, P53 will then signal the cells to die. So we thought that if we expose the stem cell to hypoxia oxygenation, surely all the stem cells should die because they're going to all activate P53. But yet, the P53 and MGM today have a negative feedback loop. There's an oscillation between P53 and MGM2. That oscillation amplitude decides whether the cell should die or survive. So we conceived that through our Sakavada Tarka, application that maybe altruistic stem cell will manipulate P53 MGM2 oscillation. And some of the cells might deficient P53 transiently for a certain period of time. And this P53 deficient stem cell, we should focus on them, identify them, isolate them, and look for altruism. So this was our experimental design. As you know, students, we need to apply logic to design scientific experiment. So far in Western science, we're using Western logic. We're the first lab in the world to use actually Indian logic. Vedic philosophical logic, Satavada Torko, design experiment, prove it. I'm going to show the data. With this modified experimental design, we then exposed embryonic stem cells to hypoxia reoxygenation. This is the first time I'm going to spend 1 minute to explain this data. Because this data, for the first time, established that stem cell can exhibit altruism. It took us 5 years to actually complete this experiment. So let me take 1 minute here. Here you can see to your left, those are embryonic stem cell colonies. I think everybody can see this laser beam. They're full of cells. This is a healthy BG01 human embryonic stem cell colonies. When you expose them to hypoxia reoxygenation, almost all cells are dying or floating, nothing almost left. And then you can see, when you take these cells, do ELISA, you can see that DCFHDA is a marker of reactive oxygen species, is highly upregulated. The P53 is upregulated. HIF was initially upregulated, HIF1, then it downregulated because it's a transient gene. HIF2 alpha is highly upregulated. When you do a flow cytometry analysis of these surviving cells, so here is this the control cells. SSEA3 is a marker of undifferentiated human embryonic stem cells. It's highly expressing here. You can see here. And then, can I take a speaker here to go there? Is it on? Hello? Hello? Yeah. So, as you can see here, SSEA3, ABCG2, there is almost no ABCG2 population in the control cells. Only few, 2%. In these cells, after exposing to extreme hypoxia oxygenation, only 8.5% cells are ABCG2 positive and SSE3 positive. When you take these cells with immunofluorescence, they have P53 deficient compared to this ABCG2 negative cell, high HIF2 alpha. When you do this ELISA, these cells here, continuously for 2 weeks, they have P53 deficiency. HIF2 alpha is up. And when you did this P53 MDM2 oscillation, as you can see, we have lots of data for this paper, but I'm only showing here the schematic. There is oscillation is completely altered here during this period. After 2 weeks, oscillation come back. When this oscillation come back, these cells die because they activate P53. During this time of 2 weeks, this embryonic stem cell secrete glutathione, antioxidant glutathione, also squalene, and that protected the neighboring stem cells from reactive oxygen species. So this is how we have published this work in Stem Cells, the leading journal at that time, and they allowed us to use this stem cell altruism because we have been able to show a mechanism, a molecular mechanism of altruism. It took us a long time, but this is our final kind of a slide to show in vitro model of altruism. A stem cell colony exposed to reactive oxygen species almost all die. Some of the cells then sacrifice their fitness by altering P53 MDM2. Eventually they die, but they help to re-establish the colony. Importantly, the self-identity. This is very important for Jiva Upakara Tantra philosophy. In a Jiva Upakara Tantra philosophy, or Indian philosophy, identity or ahamkara is the key for self. So as per that, any kind of cells, clone, should have their own identity. So it seems that our stem cell having a colony could have identity, self-identity they develop. So that's why one of these members was able to sacrifice its fitness for the rest of the community. I will talk about this self-identity later on. So this was sufficient to have a press release by Stanford University because it was a big discovery at that time, not for us, for entire scientific world, because somehow we found a molecular mechanism that can support Darwin's theory of group selection. So with that, we have this, the defending power has been established. So now how many minutes I have? Another 10 minutes? Oh, thank you. So you see, we have calculated this defending power. So we have modified Hamilton's theory of altruism. And I'm going to skip this one, go for another discovery we made. We then hypothesize that if stem cell altruism is a potent regenerative force, a potent healing force, bacteria will certainly exploit it in our body. And one of this always a very important thing, public health problem in India, is a tuberculosis. Tuberculosis bacteria hide in our body. We do not know where. As you know, if somebody has tuberculosis, we have to treat them for 9 months. Compare that to any other having an antibiotic, infection in lung, anywhere. Maximum 2 weeks of antibiotic course, doctor will give you, not 9 months. Why we need 9 months for TB? Because somewhere the bacteria hide. I thought bacteria may be hiding in the stem cells. So then we were the first one to report that bacteria can hide inside stem cells. Here's a nice picture of that. You can see those are the mesenchymal stem cells. We published this is in a Science Translational Medicine back in 2013. Those are the bacteria hiding inside stem cells. This was very not even believable because in our stem cell biology field, we thought bacteria cannot go inside stem cell. If the bacteria invade stem cells, the stem cell will differentiate because stem cell would not tolerate that kind of toxic environment. But somehow, there's a crosstalk between bacteria and stem cells. And so far, many laboratories in the world are actually actively looking for the crosstalk between microbes and stem cells. So since then, we have a series of publications. We have developed different models of studying the stem cell altruism. I would not talk about that. That was my initial theme back in 2003 in Hospital for Sick Children, Toronto, where I did my clinical research fellowship on medical oncology. Then I did my PhD in oncology. And then we moved to India here. This story began here in 2010. As a part of mandate from my father from Mumbai, I was supposed to come back to India to establish this altruistic stem cell field right in India. So I came back in 2010. We have Vijay is here, Dr. Istapoulos here. With the Gates Foundation grant, we came to Rohing, Arunachal Pradesh nearby. We set up a temporary stem cell lab where actually we had the key discovery. We have Dr. Istapoulos sitting here, right here is a co-author, Vijay Samiji too. That was as per the mandate from Mumbai Abdal Krishnabura and my father, late father, that as soon as we discover altruistic stem cell wherever in America, you should come back and get the data from India and bring Indian team here. We fulfill that by working with Rewatch, an organization from Arunachal Pradesh. So with that mandate, we also have this series of students who are taking forward this stem cell altruism field right here in Assam. I'm going to talk about them one by one for the next 10 minutes, if I get time. So first is Jyoti. She showed back in 2016-17 that how cancer stem cell manipulate mesenchymal stem cell. If you take the conditioned media of cancer cells and treat the mesenchymal stem cell exhibit altruism. So cancer is already exploiting this altruistic stem cell for their own benefit. This work is like here, you can see this after this conditioned media treatment. We can see CD271 positive, CD45 negative stem cells upregulated here, and this exhibit the p53 deficiency. I would not talk about that because we're still working on this manuscript. We're still gathering clinical data. Jyoti has completed her PhD, completed her postdoc, because we need some important clinical data. We had difficulty in funding. Because ICMR DBT, they do not understand what we do. You write a grant to them, they say, no innovation. No innovation. We don't get grant. Whatever, we have been trying to do this. So it shows when you're doing science, you have to stick to your idea. It might take years to publish one paper. You should not give up. Continue. So hopefully it will be done. But Jyoti did something great. She worked with Rupam to publish a paper on Jiva Upakara Tantra. We applied Jiva Upakara Tantra philosophy to argue for embryo editing, CRISPR. This is a peer-reviewed publication coming from Stanford University, first showing Hindu bioethics view on embryo editing. We're applying this Kamrup or Jiva Upakara Tantra philosophy here. So this is no longer an oral tradition. Jiva Upakara Tantra or Vedic altruism has been a scientifically peer-reviewed now theory or thought. Thanks to Jyoti and Rupam and Lipika. We have Sonda has done PhD on our 3 decades of the community-based participatory action research that we started in 1994, and she worked with herbal medicine. We have identified about 32 unique herbal plants from our Kamrup area. She had an ICMR fellowship on that grant as well. Thanks to ICMR for that. Then we have Sonda Jyoti has been working to find out the big bang of cancer. What about those stem cells, altruistic stem cells? They are P53 deficient, right? What about if they transform to cancer cell? Because cancer is all about deficient P53. So we develop a model here. We have taken the 4NQ carcinogen. It's a very well-known model of oral carcinogenesis. Using this model, we have found a clone that's P53 deficient altruistic stem cell. They become the cancer stem cell. This is still under review. We're still collecting data because we have not been able to confirm the transformation of the altruistic stem cell to cancer stem cell. It's another example, it takes years to demonstrate one finding because we're working on stem cell altruism. It's a very controversial field. You need data after data for that. But hopefully it will be published soon. Then we have Seema also used the same mouse model to show that mesenchymal stem cell altruism can actually lead to immune suppression. So cancer may be using this stem cell altruism to defy our immunotherapy as part of this work. This is still ongoing work. Also, Vidisha has been working on a microfluidic model to further identify altruistic stem cell in our circulation. So we can demonstrate about the role of altruistic stem cell in our healing or in disease. Back to this one. Do you have any conclusive data to show cancer is a cooperative system or not? For next 5 minutes, I will show data. Our publication mainly. First one is this. We published in Cancer Research in 2019. We showed the MYC-HIF2 pathway, the pathway that was activated in altruistic embryonic stem cells, HIF2 alpha, is again activated in a mouse model of cancer. And this mouse model was developed at Stanford by Michael Bishop, who won Nobel Prize for that. I used the same mouse model to identify cancer stem cell with my mentor, Dean Felser at Stanford. We showed in that mouse model, there is a cancer stem cell that exhibit altruism and they activate this MYC-HIF2 stem pathway. This pathway is very important for pharmacological students and scientists. It could be used for drug development because we are the first to show MYC-HIF2 alpha transcription factor that kind of cooperate with NANOX, SOX2, OCT4. Those are core embryonic stemness factor. So eventually we found this pathway in those numerous publications where HIF2 suppress p53. At the same time, HIF2 activate NANOC, sucks to for transcription factor that transforms stemness, quiescent stemness to altruistic stemness. And if the MYC is activated with p53 suppression, some of these stemness can be transformed to altruistic tumor stemness. So then we published in 2019, oral microbiome is how it manipulates cancer to exhibit altruism. And then came Dr. Lipika Pathak, she was our next student. With her, we showed that mesenchymal stem cell in the lung can be manipulated by Mycobacterium tuberculosis during the COVID infection. So we then predicted that after COVID epidemic, there will be a surge of increase of TB reactivation. This is exactly what happened. So I'm not going to go details here, but this work that we have shown how COVID-19 bacteria and this altruistic stem cell could be used for new vaccine development became a press release back in 2021. It was published by Elsevier, and for the first time they accepted this study of stem cell altruism was inspired by the Indian Vedantic philosophical theory of altruistic behavior. Proving altruism in mammalian cell biology is a challenging task as we faced years of resistance from colleagues. Commander Bikul does. Altruism is about group selection, which is very challenging to demonstrate at the molecular level. However, the idea of altruism that we are describing here is the philosophical view of Vedic Jiva Upakara, Vedic altruism, a part of Vedantic thought that states that during stress, the living organism acquires a higher state known as avatar. This is for the first time a scientific group has accepted an Indian philosophy has been transformed to scientific hypothesis and finding has been documented. Thanks to our ancestors that now we can apply our philosophy from Kamrup, India for scientific experimental design. So then we have been using this altruism to target cancer stem cell. It was published in 2022 in Translational Immunology by Seema, our another PhD student. She is now completed her PhD and she was working on Excel Healthcare in Guwahati. Now she's back to her lab. And then we finally showed in last year, we have been able to isolate altruistic cancer stem cell from my patients whom I treating, who have been treated with cisplatin. As I showed you back in 2003, that in vitro when we treat hematopoietic stem cells, other cells, with cisplatin, we could find altruism. So we have been able to show that finding clinically in 2025. 25 years, you might take some time. So this is now well documented that we can identify altruistic cancer stem cell from our patient treated with cisplatin chemotherapy. This data shows that it may be actually beneficial that to kind of select our patient whom to give cisplatin chemotherapy, whom not. In oral cancer or head and neck cancer, cisplatin is a gold standard therapy for stage 3B and stage 4 cancer patients, where surgery is no work, radiation has no use. But only 60% time cisplatin work. 40% cisplatin is worst. We found a mechanism. Now we could use this circulating altruistic cancer stem cell to decide whether the patient should get cisplatin or not. We're now clinically using this knowledge while treating cancer patients. This is an example in India that how we can do back to bedside research, back to clinics. As a clinician, this is a happy moment for us that from our lab, we have produced some science for applying in our clinics. Hopefully with the support from Indian government, we can do a large-scale trial on that and use this science and show to the world that we can bring novel Indian idea to the world stage in the terms of application of stem cell altruism in medical oncology. This is some idea. The data from here, this is the identification of altruistic cancer stem cell. These are all the molecular markers identified. They're hypoxic in the circulation as shown by the tumor result staining, hypoxia marker. Also here in the primary tumor from my cancer patients, I also found those cells we've identified. And then after that, our Partha is sitting here. We have been showing how bacteria manipulate altruism in the cancer cells and how altruistic cancer stem cells manipulate bacteria. I have 2 more minutes. In India, 90% of our cancer patients die, not of cancer. They die of infection. They die of septicemia. Because cancer patients become highly vulnerable for bacterial infection. I always ask why. I always thought that bacteria may be manipulated by these cancer cells. Maybe bacteria has been made more virulent by cancer cells. This is what we found. This was the mechanism here. We have a review paper, those who want to read. And ZBC is now reviewing our Partha's paper on that, this big finding, how the altruistic cancer stem cells are manipulating the bacteria and bacteria is manipulating the cancer cell. Crosstalk of altruism between bacteria and our cancer cell. With this, Sadhara Torko is the main result for us, for our philosophical view, for holistic view. This Torko has been immensely useful for us, and we always welcome our scientific students to use this Torko for scientific reasoning. This is the summary. We have demonstrated, we have identified altruistic cancer stem cell, positioning that cancer could be a system of cooperation, not always Darwin selection or competition. Thank you, KaviKrishna Laboratory, and this is our thorough lab where we are doing this microfluidic device and biosensor. Thank you so much. Thank you.
Before actually thanking, I think I should congratulate you for this Vedic proving our Indian logic, using our Indian logic for proving the Western logic, and this new concept of altruistic stem cell therapy for cancer. It's a very completely new idea. We have been working for some time with cancer, but it's a completely new idea that we are observing. Even in Israel, I have seen people working on stem cells regularly, but in India we are lagging behind in stem cells, particularly because of the regulatory is still not completely fulfilled. So I must really congratulate the efforts you are taking. And this whole new concept of hypoxia and oscillations can actually induce P53. It's a new thing actually for us. So we hope this will be a very helpful and it will give a new solution for how we look on cancer therapy. And we are very much thankful for your wonderful speech. My privilege to share this chair on this dais with you. Thank you very much, sir.